Hey, what's going on? It is January 9th, 11:15 right now. And welcome to your daily immune system slash eternal life learnings course. Okay, so yesterday I didn't get to post a video. I was in the middle of it, recording it, and I kind of got sidetracked. But I was thinking, so I wanted to go more into the T cells today because a little bit more about the T cell maturation, because I've been talking about the immune system. I haven't been going super deep, and I'm kind of jumping around because I'm still processing this information and building connections in my neurons, etc. Right? But one thing we should talk about eventually is what happens. How do these cells have become aware of the antigens, the antigens they need to fight to understand, so they can fight pathogenic infection, right? So there is a process for that, and it's actually fairly involved. But what I will say, and it's all specifically for T cells, it's all because of T cell selection in the thymus. It's because the thymus is where T cells mature, right? But let's talk about, before we jump into that, let's just quickly talk about the different receptors. T cell receptors. Okay, so because there's T cell receptors, there's different kinds of, we talked about B cell receptors yesterday, right? But T cells also have receptors, which they use to differentiate. They use for several reasons, similar to what B cells receptors do, but T cell receptors are used so that they can differentiate between sick and healthy cells in the case of cytotoxic cells, right? Because cytotoxic cells attack things like cancer cells, right? Because they can use the T cell receptor to determine from their MHC class 1s whether the cell is a cancer cell, because they will look at the target peptide and see if there's been mutations to it, and they will then destroy the cell based on that, right? So there is, remember, on the B cells, we talked about the B cell receptors being antibodies, and at the tips of the B cell receptors, there's something called somatic hypermutation, right? And somatic hypermutation occurs in this region here. And if you just imagine what the B cell receptor looked like, it occurred in the region at the tips of the receptor, and they really are just a process under which the receptors themselves for the antigens change so that they can recognize the genetics of that individual somatic cell change, so that the receptors that they present can differentiate between different antigens and essentially adapt, which is part of your adaptive immunity. Okay, so one thing we want to know about these receptors is that there's two kinds of T cell receptors, and T cell receptors, they're usually for the helper cells. They usually fall into the category of the alpha, beta, or gamma delta types, and the Greek lettering refers to the subunits, right? Here's the alpha subunit and beta subunit for the alpha beta T cell receptor. Okay, there's also the gamma delta T cell receptor, which is apparently a much less understood T cell that kind of floats around near your epithelial regions, or sort of close to the surfaces or interfaces of your body. So the reason for that, in the case of the gamma delta receptor, is that these are kind of like your first line defense cells. There's lots of first line defense cells like macrophages and things like that, but these T cells also provide first line defensive mechanisms, and they assist the macrophages just ingest. They just ingest pathogens like viruses and fungal myotoxic agents and entire bacteria, right? And other types of smaller types of cellular parasites, I guess. Eosinophils are responsible for bigger ones, but the T cells also do that. The T cells also lyse cells. They're these first line gamma delta T cells attack cells, and before they even get too deep within your body and attack antigens and pathogens. But they're apparently they're in the epithelial regions. They're still not well understood, which I don't know why yet. I haven't looked into that, because it seems like it would be easier to study your cells that are closer to your skin interfaces and other parts of your body. But that's a topic for another day. Okay, so back to the alpha beta T cells. The alpha beta T cells are also, the T cell receptors have co-receptors. Remember we talked about, I briefly mentioned CD8 and CD4, the two major co-receptors of T cells. So remember that CD8 is associated with your killer T cells or cytotoxic T cells, right? And CD4 is associated with your T helper cells. Okay, and they both kind of work together to bind the antigens. All right, so if you have your, they kind of like live next to the receptors, right? And they kind of cluster together, right? So they're called cluster differentiation factors, right? So basically what happens is, this is kind of like what essentially happens for a killer cell, a T cell mediated killing of an infective cell. So they attach, they then send this porphyrin protein through, and then that's a whole different topic, but that's essentially how that occurs, right? And the CD4 cells, remember, those are more for activating the B cells, right, or the weapons factory cells, if you want an analogy. Okay, so that's kind of what happens with the T cell receptors in short. There's a ton that goes on, including cytokine and interleukin interactions, and what happens once the activations occur. But let's jump back to the thymus. So the interesting part about all this is that remember, there's multiple different types of T cells, and T stands for thymus, right? So, it's kind of shorthand for thymus. So how do these T cells kind of turn into what they need to turn into, right? So T cells originate in the bone marrow. Okay, so B cells are originating in the bone marrow, T cells originate in the bone marrow. T cells, though, leave the bone marrow eventually, and they go into your bloodstream, and they float all the way into the thymus. Okay, so the thymus has specific ways that the T cells get into the thymus, right, through these entry and exit regions called HEVs. And then what happens is they go through, and I've seen an analogy for this I thought was really interesting. They go through a battery of tests, right, to determine through their maturation stage whether these cells should be CD4, CD8, or positive, or T regulatory cells. Okay, because these are the three major flavors of T cells that are interacting in your system, right? And you can think of these thymic cells, antigen-presenting thymic cells, as like bootcamp instructors, and you can think of these T cells as the recruits. So the T cells get there, and this is kind of brutal actually, right? The T cells get there, if you think about it, and then they have to first build their receptors, right? They go through stages of this selection process, right, called T cell selection. They have to build the receptors. Once they build their receptor, then they have to test whether they are able to interact with MHCs, molecules. Okay, so at first T cells are both express both CD8 and CD4 glycoproteins. Okay, so both of these are there, and they help in recognition, MHC recognition, major histocompatibility complex recognition. So they have to weakly react with the MHC. So it's basically, the first test is a test of whether the enemy can be recognized, right? And so once that's the case, then first these T cells, which are, before they go through any test, they're called double negative T cells, right? Then they go through their first test, and they become the second stage T cells, like single positive, single negative. Okay, so that means if they pass, right, if they can recognize on their MHCs the antigen, right, that the thymus needs them to recognize, then they can function in that capacity and they pass. If they don't, then right off the bat they get culled and they get apoptosized by the Thymus cell, who sends out a cytokine called FAS. Right, FAS interacts with the T cell and causes it to kill itself. So it's basically like a drill sergeant saying, you're worthless, you're worth nothing, and you don't deserve to live. And then the recruit, being so immature, decides to off themselves. That's pretty dark, but that's essentially what happens, right? Who said that nature wasn't brutal? The second stage of selection is the self-recognition. So you can recognize the enemy, but can you avoid killing yourself, right? And that's really important, because if T cells were attacking non-pathogenic tissue, then that is the source of autoimmune disease, right? Autoimmune, like self-immune disease, right? So if T cells, so the second test is that the thymic Bootcamp cells, and this is occurring in different regions of the thymus, by the way. And so it's kind of like, kind of like Squid Game in a way, right? So these thymic cells they put out a self-antigen, okay, something that your own body produces. Now, if the T cell fails to recognize the MHC class 1 or fails to bind to the MHC class 1 when and class 2, when the self-antigen is present, then the T cell is good. And it's like, oh, well, you're not going to attack yourself, you're not going to attack the body, the thing that gives you life, so you go on to your next gig. And if, again, the T cell does not recognize the antigen, or does recognize the antigen, that means there's a chance that it could attack itself, and we can't have that. So then the bootcamp instructor yells at the T cell through the use of this fast apoptizing cytokinetic interaction, and the T cell is like, oh, well, I should kill myself because I'll just end up hurting my friends, and I don't want to do that. So there you go, friends and family. So there you go. So after that, then the T cell is safe. The T cell is safe, but it still has to then, the surviving cells are self-tolerant, right? So that's what happens here. Aggressive and negative, they're double positive, and then afterwards is when the T cells have to kind of be assigned a job. So the assignment of the job is random. The T cell can be turned into either just by random chance, can either be turned into a T helper cell or can be turned into a T killer cell, right? So I like to think about it as if you're aggressive enough, you'll become a CD8 cell, which is a cytotoxic C cell, and that involves you right in the attacking of pathogens. Or if you want to set up, if you're more technically inclined, you'll be one of the engineers or scientists, or but still in the field. You're still soldiers. The T cells are still soldiers. They will still be in the field and helping the B cells arm themselves, right, and produce antibodies, and helping other cells like macrophages figure out where to go. So, that's basically what happens, right? And then there is the situation when T cells become T regulatory or T suppressor cells, but that's a whole different situation there. But that is one of the other paths that the T cell can take once it matures outside of the thymus. So just wanted to go over that. I thought it was super interesting, and this is going kind of long, and I'm going to break these pieces down in future lectures into smaller pieces so that we can really understand how T cells work and how they work in the immune system. We'll go back though to an easier topic next time. All right, thanks for listening to my chatter. See ya.